Okay, Bryn, can you hear me?
Yep, I can hear you.
Excellent. Well, thank you for joining us today. And thank you, those of you joining us through YouTube or Facebook or Research Quest Live. My name is Madeline Larson, and I'm here today with NHMU mycologist, Dr. Bryn Dentinger. Bryn, I always say, is it the G the way I said it, or is it a harder G?
That's correct. Yep.
Dentinger. Well, thank you for joining us here today. I am just getting my feet wet a little bit on our Zoom platform and feeding out into these areas. So I want to start by remembering for anybody out there tuning in to our Research Quest Live Q&A with Dr. Dentinger, that there is an Ask a Question button at our researchquestlive.org website, and you can send in your questions for Bryn, and he will be here for a half an hour and answer your questions and some of ours. So please don't be shy. Join us in asking Bryn some questions about his work and any burning questions you have about fungi or mushrooms. Let's start there, Bryn. Can you help us to know what's the difference between a mushroom and is it fungi or fungi?
Well, we tend to use the word fungi to mean all organisms within the kingdom fungi. Although the term can be used rather loosely, we sort of distinguish between an ecological definition for the term fungi or an evolutionary definition for the term fungi. The former applies to lots of organisms that don't all share a single common ancestor. So things like slime molds, we now know are not actually part of kingdom fungi. So we could still refer to them as fungi in the ecological sense. They sort of act like true fungi, but they're not part of the evolutionary sense of the term fungi. Now, mushrooms are just one group within the evolutionary group fungi. So not all fungi are mushrooms, but all mushrooms are fungi.
I see. And you study all of them.
Yeah, I focus on the mushrooms. That's my area of expertise, but all fungi are part of our domain as mycologists.
Okay, and so did what happened to Pluto basically happen to slime, and where did it go?
So it got reclassified. So fungi originally, if you don't know, were considered to be plants. And then in the 1960s, that view changed when people realized that really the way that they feed is so fundamentally different than the way that plants get their energy, that they ought to be segregated and classified in their own group. And then we've been able to refine that a little bit more, particularly with molecular data, DNA sequence data. And that has enabled us to really reconstruct the family tree of life with a lot of accuracy and resolution. And that has resulted in things like the slime molds, the water molds, which are common plant pathogens. Those turned out that they actually don't share a most recent common ancestor with all the other fungi, but with other organisms. In terms of water molds, they're more closely related to kelp than they are true fungi. And the slime molds are sort of on their own branch, kind of near the true fungi, but they diverged before fungi and animals diverged from each other.
Fascinating.
So we're constantly refining these things. This is a it's a stepwise process, and in every year we're learning new things about the history of life, and we're able to then refine our family tree a little bit more.
Excellent. We hear that a lot, don't we? Science is always evolving, it's changing. Our understandings, our research takes us to new questions and new understandings. I know in Research Quest, we always encourage our students to have flexible thinking and to follow the evidence. And so sometimes the evidence takes us in new directions. Plus slime, boy, that sounds like the subject of many a good horror film.
Well, yeah, in fact, people regularly call into their local police stations asking them what this alien thing on their lawn is, because they're fairly common in parts of North America, And people will walk outside and see like a gigantic yellow slime on their lawn and think it is actually alien. What's kind of cool, they move around. If you were to watch them carefully for a period of time, you'd actually see them migrate around.
I think I'd be okay not to see that in my yard.
They're called the fungus animals for that reason, mycetozoans.
Oh my goodness. I was thinking of you the other day because under my blueberries, I have a bajillion mushrooms right now that I haven't seen in my yard before. And I wanted to try to ID them first before I sent you a picture, but they're just everywhere. So you noted that you focus your research on mushrooms, but I was also told by a mutual friend that you're very interested in Dracula orchids.
Oh,
And
yeah.
so I'm interested in what they are and why you were interested in studying Dracula orchids.
So Dracula orchids, Dracula is the genus of these orchids, and they live in South America and Central America in the mountains. They like it rather cool but very wet. And what fascinated me about them is they produce flowers that look and smell like mushrooms, and they really look like a mushroom. They would be someone unfamiliar with mushrooms might see that and think it actually is a mushroom. They're that good at mimicking mushrooms. And so I was interested in what was actually going on here. Is it a true mimicry, or did they just evolve this morphology convergently? Is it by accident that they look that way? And so my team and I went down to Ecuador to a cloud forest and did field research on these orchids. And we were able to identify the pollinators of the orchids and found out that those pollinators, they're little flies, like fruit flies, in the genus Zygothrica. And these fruit flies, or fruit fly relatives, are all obligate mycophiles, if you will. So what that means is they all require mushrooms to complete their life cycle. Now, we don't exactly know what they're doing. One thing that we know they rely on is the male flies will aggregate on the caps of mushrooms and use them as lek sites. So they'll get together as a group and they'll battle each other for areas on the mushroom cap, and then they do these dances to attract females, and that's how they will find a mate and be able to reproduce. And so that part of their life cycle, because it takes place on a mushroom, is essential. And what we think is happening then is the orchids have exploited that by making their own mushrooms for the flies to aggregate on. And when they do that, they accidentally pollinate the flower.
Oh, fascinating. It seems like as good a theory as any, right? I mean, that's absolutely fascinating. And I'm really intrigued by this phenomenon of mimicry anyway in ecosystems. So that's fantastic. And you certainly study other things. What are some other core areas of your research that you can share with us?
So I do a lot of just fungal diversity surveys, again, focusing mostly on mushrooms around the world. So I've done lots of surveys around the world, most recently in Cameroon, in Central Africa. And that research has uncovered about 200 species or so in a very small area of forest. And probably half or more of those are new to science. So that's kind of the thing that I and many of my colleagues regularly deal with all the time, is this massive amount of unknown diversity. And so we have to sort of sort through it all and make sense of it all. And that's, I would say, the bulk of my research is focused on that aspect. And then on the other hand, we're interested in what they're doing and how they're doing what they're doing. And that involves looking into genetics. So one thing that we're looking into is the genetics underlying the biochemical synthesis of compounds, chemicals that the fungi make. These might be defense compounds, or they might be compounds used for other things other than primary metabolism. And we're interested in knowing how they actually manufacture them with enzymes, because we want to know, we want potentially to be able to exploit these for our own benefit. So we could create new antibiotic drugs, for example, by harnessing leveraging the biosynthesis capacity of fungi in nature. So to understand sort of the nitty gritty of how that all happens is another sort of area of focus in my lab.
Are there examples that you have of medicines or treatments that have leveraged that biosynthesis in mushrooms? I know that on campus, we have a professor who had been studying the toxins in cone snails for pain relief. And I'm curious if there's other research, if there's research about that can serve as an example for ways that those biosynthetics are used in medicine.
Well, it's rather early days yet for being able to harness some of these biosynthetic gene clusters, we call them, from mushrooms. But there are lots of promising leads. The one that's most prominent and one that I'm actually working on is the gene cluster that creates the enzymes that makes psilocybin, the drug psilocybin. Now, this is the active drug in so-called magic mushrooms. This is the psychoactive substance, and it's been shown to have, recent research has really shown it has a powerful potential to treat severe depression and anxiety and PTSD and some other things more than any other antidepressant ever has. So it's really a promising area of research for psychiatry. And unfortunately, it's difficult to manufacture synthetically, but the fungi can do it really well. So if we can harness the power of the fungus to do it for us, we can then start to not only make it more cost effectively, but we can also start manipulating it to produce new drugs that might be have an even higher therapeutic potential. So that's one example.
Well, I'm looking forward to more in that frontier. Let's turn to a few questions. So today we were in our Research Quest live session, we were studying the trophic levels of organisms, and mushrooms exist at the decomposer level. And you mentioned at the start of this that some people think they're plants. And Luke from Layton actually wondered, why are mushrooms not considered plants?
So they're not considered plants because they don't share a recent common ancestor with plants. In fact, they share a recent common ancestor with animals, it turns out. So even though they sort of look and behave like a plant, they're sedentary, for instance. They reproduce by spores that are dispersed aerially by wind. Those are features that you would normally associate with plants, but those are fairly superficial features, and there's only a handful of them. The rest of the features that we know about the fungi are actually much more like animals. The way that they store their carbohydrates, for example, is in the same form that we do, glycogen. They have the same types of amino acids that they use for their proteins as animals. And so there's lots of more in-depth features that actually unite the fungi and animals. We also know by looking at DNA sequences, we can reconstruct the family history of these organisms, and those DNA sequences unequivocally show that fungi and animals are more closely related to each other than either is to plants. So for those reasons, we don't consider them plants any longer.
Great. Well, on behalf of Luke, thank you for that response. We were also looking at how energy and matter move through a system, and learned, obviously, that decomposers like mushrooms have the role in the system of breaking down organic matter. And one of the things we're investigating is how will changes in an ecosystem impact that production of energy and the flow of matter in a system. And I wonder, could you tell us a little bit about how mushrooms are typically impacted in a changing ecosystem, particularly a montane ecosystem?
So the short answer is we don't know. And the reason for that is we lack the baseline information about these organisms that we take for granted in plants and most animals. So we're not able to track changes because we don't know what the baseline is. So that's a fundamental problem for us. Now, I can speculate on how things might change given what we know about the biology of these organisms. And that is when you lose moisture, you're going to lose a lot of these organisms. They depend heavily on moisture. And I think probably anecdotally, most of us already know that because we know that mushrooms show up after it rains, for example. The other thing is when we see changes in nutrient profiles, like nitrogen especially, that can really alter the communities in the soils. So nitrogen is normally a limiting factor for plants, and many of the mushrooms, for example, that we find in the mountains in Utah, are not directly involved in decomposition, it turns out. They are mutualists. So they live on the roots of the plants, of the trees, and they exchange nutrients that they get from the soil for sugar that the plant makes through photosynthesis. And when that nutrient profile of the soil is altered, that then changes, it impacts the relationship between the fungus and the plant. So if we see increasing levels of nitrogen, then the plant no longer needs to pay a fungus for that service. And so those fungi will disappear. So those are the kinds of things we might expect to see.
So are you suggesting that in a nitrogen-rich environment, you would see perhaps more fungal activity than in a less nitrogen-rich environment?
It's different types of fungi will occupy those different environments. So the mutualist type that I was just talking about, those will largely disappear. Some hang on, some are actually adapted to nitrogen, it seems, high nitrogen, but most will disappear. That's why we, in our agricultural practice, we now have to apply nitrogen to the soil because we've altered the mutualism to a point where there are literally no mutualistic fungi in the soil any longer. So now we have to artificially apply that nitrogen.
Interesting. So are you saying then that fungi, that you started your answer by saying that they're not all decomposers?
That is true.
And you're meaning that they actually don't help break down organic matter. They actually exchange nutrients and serve as a mutualist in the environment. How would you classify them on the trophic and within trophic levels?
Well, that's a good question. I don't think trophic levels deal with mutualisms, with symbioses in a very direct way. I think they tend to be folded into these different classes, but really they operate sort of in their own independent trophic class, if you will. So they're not feeding on one another. They're feeding with each other, if you will.
Sounds like we have a lot still to learn about that.
Yes, oh, for sure. Yeah.
I have another viewer question. Mary from Sandy wrote in and said that she had recently put bark chips in an area of her yard. And for the first time ever, she's seeing quite a bit of a number of mushrooms that are growing in this area, and wondered, was that the bark or was that, something else that was causing this shift? And if it was the bark, how?
It probably is the bark, and it's probably for two reasons. One, it's a source of nutrition, so that bark is not yet decomposed, and filamentous fungi are the primary decomposers of that kind of material in nature. So they're going to be the first things that you see with a fresh application of bark. That they will disappear eventually as those nutrients get depleted, and then you get a turnover into different communities of organisms that then break down products. So that's one. It's just simply a source of food. The other is that the bark probably holds a little bit more moisture underneath it, and that is good for fungal growth. So it's sort of a combination effect.
Great, thank you. Earlier, in some of your earlier responses, you were talking about some of these fascinating places that you study around the world. And I wonder if you can tell us about some of your favorite field sites around the world and what you like best about studying fungi in those areas. Fungi.
Well, I love that question. So behind me, you can see these beautiful turquoise colored or I don't know how to describe that color exactly. Mushrooms. These are, this is a new species that we uncovered in Cameroon, in Central Africa. And that has been a really fascinating place to work, mostly because the fungi there are virtually unknown. There's never really been any systematic study of them. And so it's always super exciting to us to be walking around and seeing things for the first time, really. So that is a very exciting place. I think in terms of like the place I think about most is in Borneo. I've done field work there over 3 years, and I, I just find the environment, the forest there, to be totally fascinating. The forest is dominated by this really tall tree in the family Dipterocarpaceae, and those all form these mutualistic relationships with mushrooms, like our conifers do in the Uintas Mountains, and that supports a large diversity of large fleshy mushrooms. And so it's a fascinating place. It's really fascinating also to be working in a habitat where it's mountainous, because you can move through multiple zones of habitat in a short period of time and see really drastic shifts in terms of the ecology. And that's really, I think, a fascinating experience for anyone to have. So I would say the top of my list would be Borneo, Cameroon, and then also the western slope of the Andes in Ecuador, where we did our work on the Dracula orchids. That's a very special place. It's very, very wet. Just about the wettest place I've ever been. And that just supports a really lush vegetation. All of the trees are festooned with mosses and bromeliads and orchids, and it's just really a fascinating place.
Sounds like a place that would, it would be really easy to be overwhelmed with how much activity there is, how many organisms there would be to study. How do you narrow your focus and not, end up sort of spread out all over the map? Do you go in with a question into your field work and stay strictly focused on that? Where do you leave opportunities for new discoveries?
Yeah, you always have to be open to new discoveries, I think. But often we do go in with a specific question or observation in mind, and that helps us focus. A colleague of mine, this is a skill that anybody that works in the field, but particularly in the tropics, needs to develop. And a colleague of mine once referred to it as putting on your jungle goggles, so that you only see the things that you need to see, and you can sort of eliminate all of that noise that could distract you. But that's part of what I find so fascinating too, is all that noise, that sort of intensity of life, the fecundity of these habitats is just, to me, there's nothing better on earth.
Fantastic. So I need some of those goggles for my own garden. I need gardening goggles. I go outside thinking I'm going to work on this thing or that thing, and I end up just all over the map. Because you're right, you discover, the closer you look, the more you discover, and then you wonder about it, and you want to spend time with it and figure out, maybe in my case, how to cultivate it. And so I can appreciate that. So if you ever find these commercially available, just,
Yeah, I'll let you know.
Hey, one of our other colleagues had said that I should be sure and ask you about fungus growing ants. That sounds pretty fascinating. Is that in that mutualism category again?
It is, yeah. So that's another topic that I'm researching right now. These are ants that live in the American tropics mostly, but they get as far north as Texas, Arizona, and even southern New Jersey. And they all are obligate fungus farmers. So they grow fungus as a food, and it's their sole source of food. And this is an ancient mutualism. This has been around for about 50, 60 million years. And there's over 250 species of ants now that cultivate fungi as their sole source of food. The one that people are most familiar with are the leafcutter ants. If you've ever seen a natural history documentary, you've probably seen images of leafcutter ants. These are ants that carry fresh pieces of leaves above them in their mandibles, big leaves, and they carry them to a subterranean nest. And they use that leaf then as substrate to grow their fungus crop. And so the ants are not eating the leaves directly, but simply using that to grow a fungus. that fungus then produces some specialized structures that look a little bit like oil-filled balloons, tiny little oil-filled balloons, or clusters of grapes maybe, and that's what the ants are eating primarily. Those structures are called gongolidia.
I have a whole new idea for our next research quest is a graphic novel that has fungi farmers, and they're building their own garden hoops using these leaves. I mean, we can have a whole comic strip for these fungus farming ants.
Yeah, so this is a very fascinating mutualism. There's a parallel system, it turns out, in Africa and Asia. And here there are termites that grow fungus, not ants, but termites. And they evolved this completely independently. And they grow a different type of fungus. Now, in both cases, interestingly, the fungus that the ants are cultivating is a mushroom. It's actually a couple of different types of mushrooms. And then the termites are also cultivating a mushroom, but they cultivate only one group of mushrooms in that case. But that's kind of an interesting thing, given that mushrooms are really only a small part of all fungi. The fact that both of these systems arrived at cultivating a mushroom might indicate something about the flexibility or usefulness of mushrooms for their purposes. That's just speculation, but.
Certainly it would have to be in some sort of energy, right? That these organisms are getting from, so that whatever that fuel source is, or that energy type. I wonder, would that, seems like there might be some crossover with the biosynthesis activity you were talking about, that the compounds coming out of it may be very specialized.
Yes, quite possibly. And in fact, that is an area of research that we are pursuing because we know very little about the biochemistry of the system. We don't know how the ants recognize their fungus, for example. We know that they do. If you present the ants with more than one type of fungus, including the one that they cultivate, they will always choose the one that they cultivate. But we have no idea how they recognize them. So we think it's chemical, a chemical-based recognition system, but we don't know what that is. So I think you're right that the chemistry is probably a really important part of this mutualism.
Absolutely fascinating. So I have a question for you, because I don't think I've ever asked you this when I've seen you in person, but do you have a favorite mushroom?
I get asked this a lot. I think I do, and it's porcini mushrooms. So it's not one species, but a group. Mm-hmm. These are wild collected edible mushrooms. They're widely traded, in fact. But it turns out that they're way more diverse than anybody really knew. And we didn't know the family tree. We still don't really know the family tree. So we're working on understanding that and also trying to figure out how they've gotten around the world and what kinds of traits might be driving their evolution. So we're currently working on that in my lab. That's a group of mushrooms that I've been passionate about for well over a decade.
And do they have a complex biosynthetic sort of story along with every other mushroom?
We don't know. Most likely, yes. But again, like with a lot of these topics, we don't have a lot of basic information. So we lack, until last year, we didn't have a genome, for example, of porcini. And there's never been any systematic analysis of its chemistry done. So we have to start from scratch in a lot of these cases. We have to generate that baseline information just simply in order to develop hypotheses that we can go and test. So we have to start out really at this really fundamental level in order to make progress in this group of organisms.
I have one last question for you. We're almost out of time, but I know when you came onto the museum, part of your work was to help build the museum's collection of fungi, and
however you want.
tomato.
Yeah.
And so I'm really curious, what does that involve? In terms of collecting and storing, and how is that, what are the complexities of it when we're developing our plant collections or our small mammals, or, there are processes involved, but it seems like there's this fragility and and dimensionality to mushrooms that, like, where plants can be pressed. I'm having a hard time imagining what the processes are that you rely on for building a collection. So could you tell us a little bit about how you build a mushroom collection?
Sure. So we can source specimens in more than one way. So we can get donations from other institutions, for example, to build our collection, and we've done a little bit of that. We also just go and collect ourselves and then deposit the stuff that we collect into the collection. The way that we do that is when we're in the field, we try to document them as best as we can in their fresh condition. So we'll take pictures of them. We will take very detailed notes on their features, particularly things that can't be captured in a photograph, like their smells and even tastes. And then the specimen will get dried. And we dry with a food dehydrator. So we can use low heat, like 40 degrees Celsius heat to dry them. Usually takes up overnight to 12 hours. Or we can use silica gel when we're in places where we don't have access to electricity. So a lot of my field works, for example, in Borneo and Ecuador and Cameroon, those are places where we're camping in the forest and we don't have access to electricity. So we dry with silica gel in that case. Once they're dried, they're fairly well preserved. We will bag them up to keep any moisture from getting in. Moisture is really the enemy here because they will readily absorb water. And when that happens, that makes them a food source for other fungi primarily. So molds will start to grow on them, and that will ruin the specimen. So keeping them dry is absolutely critical. When we bring them back to the museum, they have to be frozen. They get frozen for a period of time, and that's to kill any insect eggs that might have been carried with the fungus, because insects are the second source of major pest infestations in museums. So if we can freeze them before we bring them into the collection, that eliminates that as a source of infestation. So once they're dried, they then get packaged into little boxes. And we custom order tiny little boxes with lids. And we have to wrap the specimen in some acid-free tissue paper to keep them from banging around in the box. We squeeze them into the smallest box we can to eliminate any of that sort of movement that might take place. And then they get put into the collection. Of course, we're recording all of the specimen information into spreadsheets that get uploaded into online portals so that people can access the information. Those boxes get put into larger boxes, organized any way you like. We've decided at the museum to organize them taxonomically. That's a very common way of organizing things, although you don't have to do it that way. And then those boxes get stored in cabinets in the common biological dry collections room. Those are heavy metal cabinets, steel cabinets with doors that really seals them in and just provides another barrier to any potential pest infestation. And that's basically how we keep them. So they take up a lot of space because we have to put them in boxes. We can't flatten them on paper like we can plants. And that's a big disadvantage to the way that we do it. But it helps preserve their three-dimensional structures that can be useful for researchers in the future. Now, that said, I should point out that most of the morphological features that are useful for mycologists are microscopic. And so that three-dimensional structure is not the most critical part, but it's useful to help people orient where they are on the specimen. So sometimes we want to look at microscopic features that come from certain places on the mushroom, for example. And if that mushroom is all crushed, we wouldn't necessarily know where to look for those. So that's why we keep them in boxes. Now, there's a whole nother layer of complexity to the way that we curate our collections, and that is we rely so heavily on molecular data, DNA sequence data now for identifying, even just identifying our fungi, that we also have to sequence DNA from every specimen. And we're attempting to do that with our collection. Most collections haven't been able to do that, but we're trying to do that at the museum. And that allows us to build a database of DNA sequences from what's referred to as the DNA barcode region, just a small region of the genome that allows us to distinguish species. And it works in most cases. So that's a whole nother layer to our curation that most other museum collections don't have to even think about. So that was a long answer.
It was great. It actually led to at least 5 more questions, which we don't have time for today, but that just means we definitely want to have you back. And when we're able to meet back up in person, I'll certainly grill you on some of those. Your work is fascinating, and we appreciate all the contributions you make to the museum. And then I also want to thank everybody for joining us today, friends, scientists, teachers, parents. Be sure to let us know what you think about Research Quest. You can do that on our nhmu.utah.edu website on our Research Quest pages. We also invite you back tomorrow morning at 9:30 to continue part 2 of our energy and matter investigation. We have that one and one additional one this week. This is our final week of Research Quest Live. And thanks again, Bryn, for being with us.
Thank you. It's really fun. I really appreciate the opportunity.
Of course. Okay, we'll see you all soon. Bye-bye.
Bye.